working on becoming more and more automated, but in the current state, it's a big challenge. So a little step by step again, and I can maybe then go to a little detail. So leukapheresis, so taking the white blood cells of the patients. It starts there that you need to have audited medical centers that can treat that patients. So again, it's quite a burden for the quality unit you have to ramp up these centers. They need to implement it into your logistics system. They need to know how to handle that and ship. You need to be in time. You have the carrier there, so the logistics kicks in there, and they need to know how to harvest those cells. Then we have established an IT system that allows you to have an end-to-end overall survey on your cells. So it's really a global system to roll out your patients. So sometimes we have a lot of centers at the moment in Europe. The manufacturing site is in the US. So from a logistical point of view, again, quite some challenges. You have this software system that integrates them all, but at the end you have a unique identifier. You have a doer, a shipper in liquid nitrogen that you need to transport as fast as possible to the cell processing center. In the center itself, as I said, it's 22 days from vein to vein we calculate. So you have about 10 days in the cell processing facility where you manipulate the cells, expand them, and then you have to assure that you have the right cell number, the viability is correct. So you have to be in a quite exact tacted time frame that the cells are healthy and do what they should do and express the CAR T. So it's again quite a challenge here to have all these materials available. And there it's again a big difference to the traditional manufacturing. In traditional manufacturing you might have an antibody, monoclonal antibody with little stabilizers and water, like 3 to 4 excipients. And here we have about 100 different materials that go into the process. About a third of them is critical to the process and product touching. So again, it's a quite massive burden to qualify all these suppliers for the raw materials even. And these are just the raw materials. So logistics, then the raw materials, manufacturing, keeping the time plan, time is running, patient is waiting. So this is really a big difference. Again, the quality check. What do you do if the cell number is not enough? What do you do if the viability is not as registered? What do you do if you have any contamination? I mean, all this stuff is usually happening, of course, in a rare occasion in normal pharmaceutical manufacturing, but here it's always much more important for a patient. You know, it's a child is waiting, so it gives you a headache sometimes how to release a batch or to remanufacture. So quality check, again, seems to be easy, but under enormous time pressure. And at the end, cells are shipped back. Again, the chain of identity needs to be ensured. Everything needs to be documented, and back the shipper goes to the treatment center. And this is how it looks like at the end, patient material. This is the frozen bag the patient at the end receives. That is thawed and then administered to the patient. So, 17 seconds left. I made it in time. So ask Imraya, right? We need always to be in time. This is a very important picture for me because it's the wall of hope we have at Morris Plains, our flagship manufacturing site. Every light you see is a patient, and every time we treat the patient, we put on a new light. So we have now treated over 300 patients at the site, continuing. We have a new manufacturing site opened in Germany, and now I'm ramping up Japan in Kobe. So we will continually bringing up online new treatment centers to treat leukemia in the future. Thank you very much.
Yeah, thank you very much. Can we take questions? Patrick.
Thank you very much for this interesting presentation. You are targeting CD19 positive cells with the therapy. Why does a simple antibody therapy fail in this situation?
Well, I'm not the complete expert here from a science point of view, but when I was in a talk of founders of the therapy, there was always the problem that you might not be able to reach every corner of the body. So the big advantage, to my knowledge, is if you have a targeted T cell therapy, that the T cells really can access every corner, going into the brain and destroying cancer wherever it is. So there is no background where the cancer can go back. So this is I think the main advantage of the CAR T therapy.
Mm-hmm. And to which numbers do you expand these cells before? To which numbers? What number of cells do you get, and what number of cells do you reinfuse?
So there is a big variation in what we get, but we have a specification of what we should apply to the patient. If I remember correctly, it's about 10 to the 9 cells per patient, and usually we have several doses then at the end of manufacturing. So we have roughly like 3 or 5 times 10 to the 9 cells that we then can freeze in several portions. So if one fails, then we can resend another. But the problem is this is not always the case. So then it makes it more delicate to that the first dose really works.
And what are the side effects you see in those 300 patients?
Well, main side effects is cytokine release syndrome. That is basically caused by a massive destruction of the B cells and the signals of the T cells. But I think it's now much more under control, since we know that these effects can occur. So you can treat with tocilizumab, so the IL-6 inhibitor. So we have also experiences now that you have the regimen before to have the tumor burden lowered, so the B cell number lowered down. If you have a certain threshold there, we experience that the CRS is not that prominent anymore. So if you go down with the cells, so T cells do not need to destroy that many cells, the CRS is lower, but not always. So this is not completely understood why CRS still occurs sometimes.
Yeah. Maybe one last question from Bea.
If you compare to the therapy of Carl June and you now doing probably more mass, what is the big changes that you did since we heard Carl June presenting this in Basel 3 years ago?
I would say that there are not so many big changes. It's still quite similar to the Carl June's therapy. We have maybe adapted the process of how to manipulate and expand the T cells. There is tweaking and optimizing that has occurred. But basically, the main process is still the same. What we really want to do now is to automate it, because it's quite a manual process, and let's say we closed it as much as possible from what Carl June did. It's now much more closed process, and now the automation will be the next step for us.
Okay, thank you. I think we need to move on.
Okay, one point. Yeah. Carl June always said that bringing it back is the biggest danger for the infection.
Bringing back?
Bringing the T cell back.
Mm-hmm. So I mean, if you reinfuse the T cells, then there is the big...
that must be a very big challenge.
So I think the big challenge is there. If you see that the T cells grow and have an effect right away, then you know the therapy works. So, but we actually, I must say, I don't see that that's the biggest challenge. We have much more challenges before.
Okay, I think we need to move on. We need to move on. So, thank you very much, Alex. And